Hello, let's do some review for the final. Okay, taking it back to R groups and tertiary structure. Remember, this is our primary structure, beads on a string, secondary structure, helix or beta pleated sheet, tertiary structure. Okay, so this is the tertiary structure. Also, chemical interactions. Let's look at all the things going on. We have ionic bonds. We have hydrophobic interactions. We have disulfide bridges, and we have hydrogen bonds. So essentially, what we're saying is the R groups, the side chains of the amino acids, okay, contribute most to this tertiary structure. That makes sense. Can we see how that's going to make a lot of sense? So here's your options. Again, hydrogen bonds, ionic bonds, hydrophobic, and when they fold, see how they're going to fold up on this? How this chemical interaction connects to the other one is going to determine your folding. Anytime that's wrong, your fold is not going to be correct, your enzyme is not going to be correct, your protein is not going to be correct. So this is where we get sickle cell anemia and some other cystic fibrosis and some other things. In aqueous environments, the amino acids fold into proteins to avoid water. Okay, can you see that? There's your hydrophobic interactions and your hydrophobic folding. Next up, number 2, genetic code. Remember the sequence of our nucleotides in DNA and the final folding. So let's go back to this. Here's our sequence. The sequence of DNA gives you a sequence of amino acids. Those amino acids have codons. Those codons code for a particular amino acid. The order of the codons dictates the structure. Once the chain is synthesized, the specific sequence determines the amino acids in order, and that's sort of the blueprint. Remember, we said we're not going to just run away with the whole thing. It's a blueprint. It's a copy. Okay, central dogma. Single-stranded. How does the structure of mRNA allow it to serve as an intermediate between DNA and the ribosome? It's the carrier. This is the copy. You made the photocopy from back here, okay? You made a single-stranded copy to send out of the nucleus and into the ribosome. Its structure is flexible enough to exit the nucleus because it only has one strand, okay? So it's a little bit smaller. It can leave the nucleus. It does have uracil, so it has a U, and it distinguishes it from DNA. So you sort of always know it's RNA because it's got one strand and because it has a U. And again, it codes for a specific set of codons. Sorry. Specific set. Every 3 of these is going to make a codon. Okay. Why is DNA replication considered semi-conservative? So, conservative would mean there's the old one and the new one, and each time a new one binds with itself and the old one goes back to the original strand that it was with, right? If this is the original strand, it makes a copy, and then the new copy goes together and the old copy stays together. Or it's just dispersive and there's little pieces. But in this case, it is semi-conservative. There's one old and one new every time. Okay, each parent strand serves as a template for a new daughter strand. The results is 2 DNA molecules, one old and one new. And that is why it's called semi-conservative. Okay, why is DNA replicate? Oh, we already did that one. Why is it called semi-conservative? And what is the role of DNA polymerase in ensuring the accuracy of the genetic code? Okay, so DNA polymerase does 2 things. One, it's the active site, right? Here's our DNA polymerase, okay? So it's the active site. It's shaped to bind and add the correct nucleotide. So remember when we did paper PCR, we had to have DNA polymerase. And when if you did real PCR in lab, we had to have a polymerase in there. It adds the correct nucleotides. The fit of it actually gets changed to ensure that it is paired correctly. It also is a proofreader. Your DNA polymerase proofreads in the 3'. Do you remember the 3' to 5'? I haven't said that for a while. 3' to 5'. It when the incorrect base is added, the polymerase detects the mismatched geometry. It pauses and removes it and inserts the correct one. Okay, so DNA polymerase has two things that it does, two jobs. Okay, so this question has three parts, the semi-conservative and the two parts of DNA polymerase. Okay, transcription versus translation. So transcription is if I ask you to transcribe my notes, you would be writing that in English. If I ask you to translate my notes, you would be writing it in another language. That's how I remember it. Or in this case, this is an example of like from one font to another. You are moving from the language of DNA to the language of RNA, but it's the same, like you're still sort of speaking English. It's just a different font. Translation is two different languages, okay? Two different languages. The ribosome converts, now you're converting mRNA into an amino acid, right? So here you're converting, it's a little bit different. Maybe it's a different font, or maybe you write in cursive or something, okay? And that's giving you your RNA. Then look, it's all prepped and ready to leave the nucleus, and now it's going into a completely different language, which is these codons are coding for these amino acids. Okay. Epigenetics. Remember, how do epigenetic modifications such as methylation and acetylation affect gene expression without changing the DNA sequence? This is how tight it is wrapped around your histones. The methyl tags, these little methyl groups, are added directly to the molecule, and they cause your methylation causes it to be wrapped tighter. Transcription factors cannot reach the gene, and it silences it because it's wrapped so tight. Versus acetylation. So here it is like a zoom in a little bit closer. So acetylation adds acetyl groups to the lysine residues on the histone. So you don't have to remember the lysine, but it adds a acetyl group and it spreads it out. So a methyl group tightens it and turns it off, if you will. An acetyl group loosens it and allows more access. That is the grunting of a pug, just in case you were wondering. So, explain how the breakdown of glucose in animals, catabolic versus anabolic. So here, I gave you this delightful picture. So catabolism is the set of pathways that break down molecules into smaller units. Anabolism is the set of metabolic pathways that construct molecules, okay? So you usually need energy here, like some ATP, and then you will make something like glucose. This would be you have glucose and you break it down, okay? Let's look at that in terms of glucose. Okay, so catabolic is glycogen, the starch flour, remember? It breaks down to glucose, releases your energy. Versus you have glycogen, you have glucose, you add energy, and you get glycogen because you want to store it. Okay, so catabolic is your glycogen to glucose, anabolic is your glucose to glycogen, your storage. So one of them releases energy, one of them takes energy to get that stored. Okay. Explain how the breakdown of glucose in animals. Catabolic is chemically the reverse of glucose in plants. Okay, so here's the glucose synthesis in plants. So this process, again, is known as photosynthesis, and it assembles. So here is our Calvin cycle, right? This is harvesting our energy. That energy is going into the Calvin cycle with Rubisco, and that is making our glucose molecule. So the anabolic reaction here, if you're comparing and contrasting plants versus animals, is the photosynthesis is using energy to make glucose, whereas here your glycogen is breaking glucose down. Okay, so those are the two different, compare and contrast the animals and how it is chemically the reverse of glucose in plants. So we eat the glucose and use it for energy. They take the energy and make the glucose that we eat. Okay, explain how both photosynthesis and glycolysis provides electron carriers. Do you get the sense? I mean, I want you to understand that a lot of these questions are comparing and contrasting what we learned about animals to what we learned about plants, because it's just sort of the reverse. One is creating the glucose, one is breaking it down. Both of them do such similar things, in which we have photosynthesis and the light reaction, right? Which is also, what is it doing? Electron transport chain, making ATP. What? Yes. Look how that looks the same, right? It is then using that ATP and Robisco in the Calvin cycle to make a glucose molecule. Here, same thing. We are moving these electrons against their concentration gradient so they can make this ATP. We're doing the same thing here. Moving the electrons through the concentration gradient, against its concentration gradient, so it can make ATP. Same exact thing. Light strikes the photosystem, the electrons move across splitting water, and they're passed through the electron transport chain. Glycolysis in the citric acid, same thing. Okay, how does the proton gradient become established? So as the electron carriers drop off electrons at the electron transport chain, the energy is released and it's used to pump the protons. Here's the protons that we're pumping against the gradient. Here they are, all up here, across a membrane. This creates a high concentration on one side, and that battery runs this hydroelectric ATP synthase pump and creates the ATP. Okay, can you visualize that? And look, it's moving those hydrogens all the way up here. Here's where we're adding the hydrogen, NAD and then ADH. That's the hydrogen that we're pushing up there. Okay. Again, electron acceptors in photosynthesis. I'm definitely going to ask you this because I feel like we've talked about it a whole bunch of times. In photosynthesis, the final electron acceptor is this NADP, right? Right here. That's the final electron acceptor. The final electron acceptor in cellular respiration is what? Oxygen, right? So in one case, it's oxygen for cellular respiration, and in photosynthesis, it's your NADP. Okay. Metabolic regulation. What is feedback inhibition and why is it essential? So feedback inhibition is you're using the product of one to determine how to do this. So it's which the end product of the pathway inhibits an enzyme and becomes the beginning of a new pathway. It prevents a cell from wasting energy and producing more of a substance that it actually doesn't need anymore. So if you don't have a lot of glucose in your blood, you don't need insulin. You just don't need it because your blood sugar is low. But if your blood sugar is low, you are going to need some insulin. I mean, you are going to need some glucose, so you're going to have to break down your glycogen. Okay, so all of this promotes the breakdown in the pancreas and the liver, and it keeps you in metabolic homeostasis. The role of Robisco in carbon fixation. Why is this anabolic, and why is it considered the foundation of all biological food webs? Well, first of all, Robisco is the enzyme that is responsible for carbon fixation. And that is pretty important because we need glucose to eat. It takes the CO2 from the air and attaches it to the 5-carbon molecule. Right here, Robisco. Remember, when it's a molecule, Robisco is the enzyme. And it is going to move this carbon fixation cycle. It's going to move it into our molecule of glucose here. Okay? 3 molecules of CO2, Robisco, and it's going to move it along here. Without Robisco, inorganic carbon could not be converted into organic sugars that provide energy and structure for plants. Okay. That's the end of this video. See me back.